So our next speaker is also a first year student, Frieda Chan, who has completed her rotations and joined the opening lab. And she comes to us, she has various degrees in music, including a PhD, and then we decided that science was going to painting, did a postdoctoral work here at WSU, and completed rotations. Frieda.
So hi everybody, and thank you Dr. Glass for the introduction. So today I'll be presenting a paper by Hobbs and colleagues in the Pendology Lab at Harvard University. This research is about how PLZF regulates mTORC1 expression to generate a balanced spermatogonial stem cells self-renewal and differentiation. So why is spermatogenesis so important? Well, spermatogenesis is a classic model of tissue-specific stem cell biology. Spermatogonial stem cells are the only cells in the body that both self-renew and differentiate into haploid cells that contribute genes to the next generation and also is involved in sexual determination. The activity of spermatogonial stem cells lay an important foundation for spermatogenesis. And if spermatogenesis is disrupted because of stem cell inhibition, it will lead to male subfertility or infertility. According to the World Health Organization, of all the reproductive couples worldwide, 15% of them are infertile. Out of all those cases, half of which are caused by male infertility. Lowered sperm count is one of the main causes of male infertility, and it can be caused by disruption in spermatogenesis. So by understanding the mechanism and pathways within spermatogenesis, we can then start to develop therapies and cures for male infertility. So the A-single asymmetric model shows how spermatogenesis is sustained by spermatogonial stem cells. Spermatogonial stem cells can exhibit two different fates. First, spermatogonial stem cells can renew into another spermatogonial stem cell. Second, spermatogonial stem cell can transition into the transient amplifying progenitor cell, which then proceeds to become A pair, A-align 4, A-align 8, and A-align 16 cells. All these cells are considered to be undifferentiated, and they will exhibit markers such as Thy1. Once the spermatogonia enter into the A1 stage and onward, it is considered to be differentiated, and they will exhibit markers such as c-KIT, alpha-5 integrin, and CD45. So this is a cross-section of the seminiferous tubule from the testis, and you can see different stages of germ cells within the seminiferous epithelium. Right here at the basement membrane is a very special place. It is called the stem cell niche. It is a microenvironment where the stem cell and the progenitor cells reside. They're supported by somatic cells such as Sertoli cells and Leydig cells. So within this microenvironment, there are many growth factors. One of the very important and essential growth factor is GDNF, glial cell line-derived neurotrophic factor. If GDNF is absent, stem cell population will disappear. And if GDNF is overexpressed, then spermatogonial stem cells and progenitor cells will over-proliferate and lead to tumor formations. So GDNF is known to react with this receptor, GFR-alpha-1 Rad complex. Then it will lead to the phosphorylation of the PI3 kinase AKT complex and the phosphorylation of ERK-MAC kinase complex, which will then activate mTORC1, leading to the phosphorylation of downstream translational machinery such as ribosomal protein S6. So if mTORC1 is overly expressed and overly activated, it will lead to the depletion in spermatogonial stem cell population and increase in progenitor cell differentiation. mTORC1 is known to be inhibited by extracellular factors such as rompamicin and regulated by intracellular factors such as Rad1. In addition to GDNF, PLZF promyelocytic leukemia zinc finger is important to the spermatogonial stem cell maintenance. PLZF is crucial for both germ cell function and the maintenance of germ lineage. Over here is another seminiferous tubule cross-section with immunohistostaining for PLZF. You can see the cells in the basement membrane that are expressing PLZF. And in a mice that is PLZF deficient, their testis will become progressively lost in germ cells, and then it will have atrophy. In this cross-section of the seminiferous tubule from PLZF deficient mice, you can see that there's a complete lack of germ cells. So the researchers hypothesized that the GDNF-dependent spermatogenesis involves a crosstalk between PLZF and mTORC1. So within the testis, there's only about 0.0% of the population that are spermatogonial stem cells. It is very difficult to work with undifferentiated cells because of all the other germ cell population. So in order to work with undifferentiated cells, they have to isolate for undifferentiated cells. To do that, they used undifferentiated marker, positively select for them, and then negatively select for differentiated marker, such as alpha-5 integrin and CD45. After isolation of such population from both wild type and PLZF deficient cell, they found that in the red box are the isolated cells from the wild type population, and it's only about 5.41% that's undifferentiated and 3.78% from PLZF deficient cells. Then they proceed to see if these populations are actually still enriched in spermatogonial stem cells. To do that, they used a recolonization assay by re-injecting these isolated populations into busulfan-treated testis. After recolonization, they recounted all the stem cell populations that generated recolonization, and they saw that the isolated populations has a 25 times higher recolonization rate than just a whole cell germ cell population. Then they proceed to make cell line cultures of both wild-type isolated cells and PLZF deficient isolated cells. After they established the cell lines, they used Western blot to confirm that they still remain the germline qualities. They used MBH mouse VASA homolog germline marker in this Western blot, and you can see that both the cell line from wild-type cell and PLZF deficient retain the germline qualities. So after isolating and culturing the cell lines for both wild-type and PLZF deficient cells, they can then proceed with the comparative analysis. So the first thing they want to do is to find out what is happening to mTOR in PLZF deficient cells. To do that, they are measuring the phosphorylation level of ribosomal protein S6. Using flow cytometry, they were able to tell that the red line is where the PLZF deficient cells, and they saw that there's an increased amount of ribosomal protein S6 phosphorylation, indicating that there's an increase in mTORC1 activity. Then they decided, well, let's use vancomycin to inhibit mTORC1 and see if we can bring back the activity level back to wild type. And here you can see that, again, the red line indicates the PLZF deficient cell, and the mTORC1 activity level is brought back down to the wild type level. So from this, the researchers know that in PLZF deficient cells, there's an enhanced level of mTORC1 activity, which can be inhibited by vancomycin. Now the researchers wanted to know what happens to the upstream pathway of mTORC1. Again, these Western blots are just showing you that in PLZF deficient cells, there's an increase in mTORC1 activity. First, they measure the phosphorylation of AKT upstream pathway. In this Western blot and this quantification, it shows you that there is actually a decrease in phosphorylation of AKT when mTORC1 is overly activated. This is not quite what they expected, so they decided to look at another upstream pathway, the ERK phosphorylation pathway. And this is the Western blot. And you can see more clearly in the PLZF deficient cell where RPS6 is overly phosphorylated and mTORC1 activity is increased, there is again a deficiency or decrease in ERK pathway phosphorylation. So this showed the researchers that with overactivation of mTORC1 in PLZF deficient cells, it generates a negative feedback loop to the upstream pathways. So knowing that overexpression of mTORC1 will generate a negative feedback loop to decrease the upstream pathway phosphorylation, the researchers wanted to know what if they treated the cells with both GDNF and rapamycin in the absence or presence of them. So they also hypothesized that if you treat the cells with rapamycin, you can probably restore the phosphorylation of upstream pathway ERK and AKT. So here you can see that in rapamycin-treated cells, the mTORC1 activity, it doesn't matter if they're wild type or PLZF deficient, in the presence or absence of GDNF, there is an inhibition of mTORC1 activity because there's no phosphorylation of ribosomal protein S6. You can see it very clearly in this quantification. Then they measured AKT phosphorylation. You can see it in this plot and this graph. And the black ones are rapamycin treated. And you can definitely see that not only the rapamycin treated cell has recovered the AKT phosphorylation level, it actually is increased compared to wild type and non-treated cells. So the researchers from this gather that rapamycin inhibition of mTORC1 increases the upstream pathway phosphorylation and may even increase GDNF receptor expression. So they went ahead and did an experiment measuring GDNF receptor, GFR-alpha-1 expression. So the gray area here, as you can see, is the wild type control of GFR-alpha-1 expression level. Then the researchers treated the wild type cells with rapamycin to inhibit mTORC1 activity. In the green line, you see that after mTORC1 activity is inhibited, there's an increase in GFR-alpha-1 expression. And then in PLZF deficient cell where there's mTORC1 overactivation, you can see that in the red line, there's a decrease in GFR-alpha-1 expression level. The researchers then treated those cells with rapamycin to inhibit mTORC1 activity. In the blue line, you can see that after the deficient cells are treated with rapamycin to inhibit mTORC1 activity, there's an increase in AKT phosphorylation. And there's an increase in GFR-alpha-1 expression. So from this, they saw that rapamycin inhibition of mTORC1 has increased the GFR-alpha-1 expression level in both wild type and PLZF deficient cells. So knowing that the extracellular factor rapamycin can inhibit mTORC1, the researchers wanted to know the activity between, what the relationship between Rad1, the intracellular regulator of mTORC1, and PLZF deficient cells. So first, they needed to confirm that Rad1 is indeed regulating mTORC1 activity. To do that, they used an siRNA to downregulate the expression level of Rad1. And in a wild type cell, this is the phosphorylation level of RPS6 and the activity of mTORC1. And when the cells are treated with the siRNA to downregulate Rad1 expression, you can see that there's a definite increase in RPS6 phosphorylation, indicating an increase in mTORC1 activity. So from this, the researchers gather that Rad1 indeed is a regulator of mTORC1 within the cell. So now the researchers, knowing that, wants to find out what happens to Rad1 within a PLZF deficient cell. They used RT-PCR to measure the mRNA transcript level of Rad1. And they saw that within PLZF deficient cells in red, compared to wild-type cells in blue, there's a tremendous decrease in transcript level. So this means that the researchers said Rad1 is the regulator of mTORC1, and because PLZF deficiency, there's a lack of expression of Rad1. So PLZF actually interacts with Rad1 to inhibit the expression of mTORC1. So finally, the researchers wanted to know if rapamycin inhibits mTORC1 activity, will spermatogonial stem cells renewal level go up, or what will happen to it? So they treated wild-type cells with rapamycin to inhibit mTORC1 activity. They used immunohistostaining in a cross-section of seminiferous tubule. The top is treated with vehicle. The bottom is inhibited by rapamycin. In the top, the little blue dots you see all along the seminiferous tubule indicate cells that are expressing PLZF. And in the bottom, the researchers, especially in the quantification, you can see that there's an increased amount of cells that are expressing PLZF. So the researchers from this saw that with rapamycin inhibition of Rad1, there's an increased amount of cells that has PLZF expression, indicating that there's an increased amount of self-renewal within the spermatogonial stem cell population. So in summary, this is the pathway that the researchers found in wild-type cells. With PLZF expression, it leads to the expression of Rad1, which then regulates mTORC1. With regulation of mTORC1, there's a balanced spermatogonial stem cell self-renewal and differentiation. And in a cell that is PLZF deficient, there's a decreased amount of Rad1 expression, which then leads to the overactivation of mTORC1, which will decrease the expression level of GFR-alpha, GDNF receptor, and lead to the decrease in phosphorylation of upstream pathways such as AKT and ERK. This will lead to the decrease in spermatogonial stem cell population and increase in differentiation. Lastly, when a cell is treated with rapamycin inhibiting mTORC1, there will be an overexpression of GDNF receptor GFR-alpha 1. This leads to the increase in self-renewal of the stem cell population. So in conclusion, the researchers found that mTORC1 inhibition increases the spermatogonial stem cell self-renewal in both wild-type and PLZF deficient cells. They also identified a new way for PLZF to regulate the cell's maintenance by expressing Rad1 to inhibit mTORC1 activity. So with these experiments and publication, they were able to find another piece of the puzzle for the spermatogenesis mechanism and pathway. With more knowledge from this spermatogenesis pathway and mechanism, we can have further understanding into what causes male infertility or subfertility, so that maybe treatments and cures can be discovered. And here are some of my critiques of the publication. First, the researchers were not very clear on how they were able to establish the PLZF deficient cell line, because PLZF deficiency leads to the lacking of germ cells. They were not very clear on how they were able to establish it, and when they're not clear on how they established the cell line, other researchers will not be able to replicate it and continue or replicate their experiments. So secondly, after they found the PLZF mTORC network, the population they used was undifferentiated cell, which encompasses both stem cells and progenitor cells. So they need to try to find another marker that can separate out the two populations to know if the pathway works in both stem cell and progenitor cell, or just stem cell and or just progenitor cell. And lastly, there was a lack of experimental replication. Sometimes it's just one, sometimes it's twice. Probably due to the fact that PLZF deficiency line is so hard to culture. And because there's a lack of replication, there was a bit lack in quantification, which summarizes to a lacking in statistical data. So I would like to thank the Oatley lab, especially Dr. Oatley, who has guided me through the whole process, and Dr. Young, Melissa, Amy, and Ryan, and all my fellow students, Justin, Reggie, James, Brandon, Nate, Olga, Jody, Kevin, Colin, and Thomas, who helped me with the preparation process. Thank you very much for your attention, and if you have any questions, I'm happy to answer them.
Frieda, questions?
So the mTOR is kind of a sensor for energy and environmental conditions. So I know the paper wasn't about this, but what do you think, I mean, why do you think that pathway is so complicated? And what are these stem cells trying to sense before they decide whether to differentiate or not?
I think what you're asking, how mTOR is an energy sensor, and why is it so complicated, the pathway, and how a stem cell actually decides if it wants to differentiate or not by interacting inside that pathway. I'm not sure, but I believe that maybe because differentiation is such an energy expensive pathway, if the condition within the testis is not correct, it will probably decide to renew or to just differentiate, and or maybe because stem cell population is so expensive to renew. I'm not sure the energy components will have it. Does that answer your question? Yes.
So I guess GDNF and PLZF is specific to chromosomal stem cells. But I think the rest of the pathway is it conserved with other stem cells like hematopoietic stem cells or?
Yes, it is, because rapamycin actually is an antifungal medication, and it is currently being developed as a medication for tumors. So, and I also know that rapamycin inhibits T cells and B cells activation and activity, from what I read.
So yeah, my follow-up question is, so has anybody looked at how this pathway is important for maintenance differentiation in other stem cell pathways?
I know that the Pandolfi lab is actually working on that, but I'm not very sure, because right now, I think the last thing they worked on, I'm not very sure, but I know that they're working on that right now.